Welcome back to Talk Tensors, the show where we try to unpack these sometimes very complicated machine learning papers and make them accessible.
Pull out the golden nuggets of knowledge.
Exactly. And today's paper is no exception.
Definitely not.
We're diving into aggregation schemes for single vector WSI representation learning in digital pathology.
Okay, so I guess a good place to start would be like, what is a WSI?
Yeah, WSI, it stands for a whole slide image.
Uh-huh.
And it's basically like, when you see in a medical lab, they have those biopsy slides, and they're examining them under a microscope.
Yeah.
A WSI is like a really, really high-resolution digital version of that.
Mm-hmm.
So it's like, imagine taking a picture of that slide with like a gigapixel camera.
So like a ton of information.
Massive amounts of data.
But the problem is that they're so big that it's actually kind of a problem for current AI applications because they're just too large to be processed by most GPUs.
Right. It's like trying to stuff an elephant into a Mini Cooper.
Yeah, it's not going to work.
So what do? They chop it up into smaller pieces.
Exactly. They break them down into these smaller chunks called patches. So it's kind of like if you were trying to eat a giant pizza, you wouldn't try to eat the whole thing at once.
You cut it into slices.
Right, exactly.
So you break these WSIs into patches so you can actually process them. But then the question becomes, how do you take all of that information from those individual patches and like reassemble it? How do you get a meaningful understanding of the entire WSI?
So that's what this paper is all about, right? They're focusing on this idea of a single vector representation.
Yeah, so instead of having like, hundreds or thousands of different vectors, one for each patch, you have this one like super vector, like a boss vector.
The executive summary of the WSI.
Exactly. It captures the essence of the entire WSI in a single vector.
Which is a pretty amazing feat when you think about it.
Yeah. And why is this single vector representation so important? What are the benefits?
Okay, well, let's think about it. First of all, storage, right? WSIs are, as we've said, they're huge. They take up a ton of storage space, which is a big problem for like hospitals and research labs. But with a single vector representation, you can dramatically reduce the amount of storage space you need.
Makes sense, yeah. It's like compressing a huge file down to a ZIP file.
Exactly. It's all about efficiency. And it's not just about storage either.
Right.
Think about comparing two WSIs, right?
Mm.
If you have to compare them patch by patch, that could take forever. But with a single vector representation, you're just comparing two vectors, which is way faster and easier.
So it makes it easier to analyze the images too.
Oh, absolutely. You can do all sorts of things with these single vector representations. You can search for similar WSIs in a database. You can group them based on their characteristics. It opens up a lot of possibilities for research and diagnosis.
I see. So it's not just simplifying things. It's actually enabling new ways of understanding and analyzing these images.
Right. And don't forget about speed. Searching through a database of single vectors is going to be much, much faster than sifting through all that patch level data, which could be really important for like real-time applications in a clinical setting.
Yeah, like assisting a pathologist during a diagnosis where every second counts.
Exactly.
So before we get into the nitty-gritty of this paper and the different aggregation techniques they explored, can you give us a little bit of historical context? Like, what was the state of the art before this research came along?
So before this kind of like explosion of research in single vector WSI representation, there was one technique that was used in a WSI search engine called Yodixel. And it was based on this idea of the median of minimums.
Median of minimums. That sounds like a mouthful.
It is a bit of a mouthful.
Can you break that down for us? What does that actually mean?
Okay, imagine you're trying to compare two neighborhoods, right? And you want to see how similar they are based on like the types of houses in each neighborhood. So with median of minimums, you would take each house in one neighborhood and find its closest match in the other neighborhood based on whatever features you're interested in.
Okay.
And then you'd calculate the average difference between all those closest matches, and that would give you a measure of how similar the two neighborhoods are overall. So it's like finding the average closest match between two sets of things.
Ah, okay. So it's a clever workaround, but it doesn't actually give you that single vector representation.
Right. It's practical for some applications, but it has limitations. This new research really pushes things forward by looking at a whole range of new techniques to create these single vector representations.
Okay, so let's talk about those techniques. What did the researchers actually investigate?
So they looked at a bunch of different approaches. Some of them were pretty straightforward, like just averaging all the patch vectors together, or taking the vector with the maximum value. Kind of like if you were trying to describe a group of people, you could either say the average height is 5'10", or you could say the tallest person is 6'4".
Yeah, so those are like the basic building blocks.
Right, but they're not the most sophisticated methods.
Did they work very well?
Not really. The really interesting stuff comes with the more advanced techniques like deep sets, which is this really cool idea where you try to create representations that are invariant to the order of the input. So it doesn't matter what order you feed the patches in, you'll still get the same representation of the whole WSI.
Interesting. So it's about capturing the essence of the group, not the specific arrangement.
Exactly.
What other techniques did they explore?
They looked at memory networks, which are kind of like inspired by how human memory works, where it can selectively focus on certain pieces of information and ignore others.
So it remembers the important stuff and forgets the unimportant stuff.
You're right. Kind of like how you might remember the key points of a conversation, but not every single word that was said.
I like that analogy. Makes it easier to understand. And what else?
Well, there's focal attention, which builds on that idea of selective attention. And then there's a whole class of techniques based on Fisher vectors, which are basically a way of representing data using a bunch of statistics.
Okay, that sounds a little more complicated.
It is a bit more technical, but basically it's like if you were describing a city, you could just list all the landmarks, but a Fisher vector would tell you not just what landmarks are there, but also how they're distributed, how far apart they are, stuff like that. So it gives you a much richer representation of the data.
I see. So it's not just about what's there, but also how it's organized.
Exactly. And they even looked at these things called deep Fisher vectors, which are basically a more sophisticated version of regular Fisher vectors. And they came in these different flavors like sparse and binary.
Sparse and binary, what do those mean?
So a sparse vector is basically a vector where most of the elements are zero.
Yeah.
So it's a very efficient way to store information because you only have to store the non-zero elements.
So it's like a really tidy bookshelf where most of the shelves are empty.
Exactly. And a binary vector is even more extreme. It's a vector where all the elements are either 0 or 1.
Gotcha. So it's like a bookshelf where each shelf can only hold one book and the book is either there or it's not.
Perfect analogy. And these sparse and binary representations are really interesting because they address these key challenges of storage and search efficiency.
Makes sense. So they experimented with all these different techniques, but how did they actually test them? How do they know which ones worked best?
So they used 4 different cancer data sets from the Cancer Genome Atlas, focusing on bladder, breast, kidney, and colon cancers.
Okay.
And to evaluate the different techniques, they used this method called K-nearest neighbors, or KNN.
PNN, I think I vaguely remember that from my machine learning days. Can you refresh my memory?
So imagine you're at a party, right?
Uh-huh.
And you want to find the people who are most similar to you based on their interests. So KNN is like this algorithm that can help you do that. You tell it what you're interested in, and it'll find the K people who are closest to you in interest space.
So in this case, they were using KNN to find the WSIs that were most similar to a given query WSI based on their single vector representations.
Exactly. And the results were really interesting.
What did they find?
Well, it turned out that deep Fisher vector, especially the sparse and binary versions, consistently outperformed the other methods.
So deep Fisher vector is the champion.
It seems to be the winner in this particular study, yeah. What's really cool about it is that it not only gives you the single vector representation, but it also comes in these versions that are super efficient for storage and search.
So it's like, it's got it all.
It does, which is pretty exciting.
But I imagine it probably depends on the specific application, right? Like if storage is your biggest concern, you might go with the sparse version. But if speed is more important, then maybe the binary version is better.
Right. It's all about choosing the right tool for the job. But regardless of which specific technique you choose, these findings have the potential to really accelerate the development of AI tools for pathology, which is pretty amazing.
Yeah, that's a huge deal. So for our listeners who might be thinking, okay, cool research, but why should I care? Can you connect these findings back to the real world?
Well, think about it. Faster and more accurate diagnoses, more personalized treatment plans, potential breakthroughs in cancer research, all of that is within reach thanks to this kind of research.
Yeah, that's a future I can get behind. But I think it's also important to remember that this is just one step in a long journey, right?
Of course, there's still a lot of work to be done in terms of validating these techniques on larger and more diverse data sets, integrating them into real-world clinical workflows, all of that. But this research lays a really solid foundation for future innovation in the field.
Right. And speaking of innovation, this paper really got me thinking. If we can summarize a whole slide image into a single vector, what else could we summarize?
Oh, you're going there. I like it. You're about to launch into our novel notions segment.
You know it. We'll be back in a moment to explore some out there but plausible research ideas inspired by this paper.
Welcome back to the show.
So before the break, we were talking about these single vector representations and how they could be applied beyond just, individual WSIs.
It's kind of mind-blowing when you think about it.
Yeah, if we can summarize an entire high-resolution image into a single vector, what other frontiers could we conquer with this approach?
Well, one area that immediately comes to mind is genomics.
Oh, interesting. How so?
Think about trying to analyze an entire human genome, right, which contains like billions of base pairs.
Yeah.
It's a massive amount of data, even for the most powerful computers.
So could we use these single vector representation techniques to create like a summary of an entire genome?
Exactly. Instead of having to analyze all those individual genes and segments, you could have a single vector that captures the overall genomic signature of an individual.
So it's like a fingerprint of their DNA.
Yeah, a genomic fingerprint. And that could have huge implications for personalized medicine.
How so?
Well, imagine being able to tailor treatments to a patient's unique genomic profile.
So instead of a one-size-fits-all approach to medicine, we could actually have truly personalized healthcare.
Precisely. And I think the applications go way beyond just genomics.
Oh, yeah? What else are you thinking?
Well, what about proteomics, which is the study of all the proteins in an organism, or social network analysis, where you're dealing with these massive graphs of interconnected individuals?
Yeah, those are all incredibly complex systems.
They are. And in all of these fields, the ability to create compact, meaningful representations of complex data could lead to incredible breakthroughs.
It's like having a map that helps you navigate through this vast and intricate landscape of data.
Yeah, a roadmap for understanding complex systems.
And you can see the major landmarks and connections without getting lost in the weeds.
Exactly. And the implications extend beyond just data analysis. Imagine using these techniques to streamline machine learning models themselves.
Okay, I'm intrigued. How would that work?
Well, one of the biggest challenges in machine learning is the sheer volume of data that's required to train these complex models.
Right, it's like trying to teach a child a new language by giving them the entire dictionary at once.
It's overwhelming. So what if, instead of feeding the model all this raw data, we could give it a curated selection of the most important information distilled into these single vector representations?
So it's like giving the model a personalized study guide, highlighting the key concepts and connections.
Exactly. And that could potentially lead to faster training times, reduced computational costs, even improved model performance.
So it's not just about making the data more manageable. It's about actually making the models themselves more efficient and effective.
Right. It's a really exciting area of research.
Yeah, this is really opening my eyes to the true potential of this work. It's not just about improving digital pathology. It's about revolutionizing how we handle and analyze complex data across a whole range of fields.
Absolutely. And we've really only just scratched the surface of what's possible.
It's amazing to think about all the potential applications that are still waiting to be discovered. But before we get too carried away with our novel notions, I think it's important to acknowledge that this is all still, speculative at this point.
Of course, yeah. We're just brainstorming here, imagining what the future might hold.
Right. But it's fun to speculate.
Yeah.
And this paper really got me thinking about one particular question. What if, instead of representing a single image, we could create a vector that represents an entire collection of images?
Whoa, now you're talking. That's going meta. You're talking about creating a vector that captures the essence of an entire data set.
Exactly. Imagine having a single vector that represents, say, all the chest x-rays in a hospital's database, or all the satellite images of a particular region over time.
The possibilities are endless. You could compare different data sets, track how they change over time, maybe even predict future changes.
It's like having a crystal ball for big data.
Yeah, a way to see the big picture, to understand complex systems in ways that we never thought possible.
It's like having a telescope for big data. You could start to see patterns and connections that would be invisible at the individual image level.
And think about the implications for machine learning. We could train models on these dataset level representations, potentially leading to even more powerful AI systems.
Models that can understand and predict complex phenomena at a global scale.
It's mind-blowing.
Of course, there are still a lot of challenges to overcome.
Oh, definitely. How do you define the essence of an entire dataset? What features are most important to capture? How do you make sure the resulting vector is both comprehensive and concise? These are all tough questions.
Yeah, but if we can figure it out, the potential rewards are enormous.
No doubt about it. It's a challenge worth tackling.
It would open up entirely new avenues for research and discovery.
It's a really exciting prospect.
Well, on that note of boundless possibilities, I think it's time to wrap up this deep dive. This idea of like representing a whole data set as a single vector,
Yeah.
it really gets the gears turning,
Yeah, like a single vector for all the MRI scans of, say, patients with Alzheimer's.
Or all the satellite images of the Amazon rainforest over like, the past 30 years.
You could compare different data sets, see how they've changed over time.
You could maybe even predict future changes.
It's like having a crystal ball for data.
A crystal ball for big data.
Yeah, exactly. Like imagine comparing the genomic profiles of like an entire population
Or tracking the spread of a disease through a social network.
it's incredible the insights you could get.
Right. It's like having that bird's eye view of the entire data landscape.
You could spot patterns and connections that you'd never see otherwise.
And think about what that could mean for machine learning
Right. We could train models on these data set level representations.
and potentially end up with even more powerful and efficient AI systems.
Systems that could understand and predict complex phenomena on a global scale.
Pretty mind-blowing when you think about it.
It is.
But, it's important to remember that there's still a lot of challenges to overcome.
Oh, yeah, for sure. Like, how do you even define the essence of an entire data set?
Right. And what features are the most important to capture?
And how do you make sure that the resulting vector is both comprehensive and concise?
Yeah, there are a lot of tough questions to answer.
But the potential rewards are huge.
It's a challenge that I think the machine learning community is definitely ready to tackle.
Absolutely.
And I'm really excited to see what breakthroughs emerge from this kind of research.
Me too.
Well, on that note of anticipation and excitement, I think it's time to wrap up this deep dive.
Yeah, I think that's a good place to stop.
So if you enjoyed this exploration of aggregation schemes for single vector WSI representation learning in digital pathology, and especially if you enjoyed our Novel Notions segment,
Which I hope you did.
we encourage you to check out the full paper. We've provided a link in the show notes.
And as always, we'd love to hear your thoughts.
Yeah, connect with us on social media, leave a comment on our website.
Tell us what novel notions this paper sparked for you.
Until next time, keep those minds curious
Keep exploring.
and keep pushing the boundaries of what's possible with machine learning.
Thanks for tuning in to Talk Tensors.